Good evening, everyone present in today's webinar. It's a lovely time for me to be here. I'm very much privileged to be a part of this webinar organized by a Facebook group and Seva, Bharati, Mahavidyalaya, as well as DRISHTICON organization. I'm very much thankful that they've made me a part of this webinar. They've called me here, so it's my privilege, I must say. Thank you so much, Didi sir. We call him Didi sir, Debrata sir, because when I was in Darjeeling Government College, right from that time, he has seen me working with the plant tissue culture. That is why he's called me here. Because plant tissue culture is so much fascinating work, and my life with tissue culture started in Darjeeling Government College. As I was there as a PhD scholar, I was there as a women scientist and postdoctorate fellow, but all works are with tissue culture only. And today also I'll discuss with the case of orchids only, because the orchids are really fascinating. I'm very much thankful to the teachers in Darjeeling Government College also. I'm thankful to the teacher, my guide, who has taught me how to do this and who's given me all the knowledge about it, Dr. Kalyan Kumar De and Dr. Aniruddha Saha. I'm very much thankful to them. Okay, let's get back to the point. I am Ranjit Amang, as you all have come to know by now. And at present, I am in Vidyasagar College for Women, Kolkata. But before that, I was in Darjeeling Government College. So recently, I've been to that college, but still, I will definitely try plant tissue culture there also. That is something that I cannot leave with. So I have to do that also. So as with regard to today's lecture, it's a plant tissue culture and it is an intervention in biotechnology. So in general, as we all know, biotechnology is a technique or the advanced technique or the development of a technique in which we apply or we improve the technique of some of the biological processes, and that leads us or helps us for the production of new materials or the new varieties which we can use in everyday life for the betterment of the life. That could be in the field of agriculture, it could be in the field of medicinal field, or in our day-to-day life. So whatever the improvement we can bring from any of the biological process, that is our biotechnology. We're trying to improve it, but the earth has given us a pause with this pandemic right now. But at least we are virtually connected together. That is also a technology. That is also the improvement in technology. And as long as we are connected virtually, at least we can learn for the time being until and unless this pandemic gets over. And by the time it gets over, we can think of something better, development of biotechnology. So one of them is definitely plant tissue culture. So if you just see the definition, tissue culture is nothing but it's aseptic culture. That means every workout is totally free from contamination under controlled environmental conditions and desired nutritional supplement. So if we grow a tissue inside a culture tubes, that is in vitro, with our desired nutritional supplements, that is defined as a tissue culture. So, of course, this was idea. This idea started sometime in 1902 when Hivelanth pointed out the possibilities of culture of isolated tissues, and theoretically he said that all plant cells are able to give rise to a complete plant. And from then there was the story of plant tissue culture. And as the years passed, there was the cultures from embryos, root and stem tip, as you all can see in the slide, the year and the worker or the scientist who has worked against that, and the human nature. They were growing it. Now, why not we insert hormones there so that it grows in a more better way? That's again biotechnology. So the first time, the first plant growth hormone was discovered, that was indole acetic acid. That is IA, indole acetic acid. We use different plant hormones at present. We use BAP, we use GA, we use cytokinin, but still we discuss or we remember the first only always. So that is why indole acetic acid comes in the line. And later on, in 1934, vitamin B supplement as a growth supplement in the root tip. So that was there. And in 1939, there was the endless proliferation of the callus cultures was established. So as the tissue culture was carrying on, the different improvements going on, then there was the utilization of liquid endosperm or the coconut water. This is very much nutritious for the nutrient media or the plant tissue culture media, because it has lots of cytokinin. Then the discovery of cytokinin as the potent cell division factor, because once the tissue culture has started use of endosperm of coconut or the coconut milk, they found that the cell division of the plant cell is very enormous, very vigorous. That is why what is exactly helping out? It's the cytokinin which is helping out. And then the shoot apex, the growing point, all this, the root apex, they were isolated and started propagation there. Then later on, microspores of Datura and Nicotiana, and then a little deeper, that is protoplast isolation. And protoplast isolation and fusion was done in between potato and tomato. That was the first fusion, so the pomato. And in 1983, that was the milestone when plant tissue culture was given a very big success, because insertion of a foreign gene to the plasmid vector and then insert into the another naked plant protoplast. So after that, what we're getting the desired plant with the desired trait. That means the desired gene has been inserted there. So that was the main milestone in plant tissue culture. Now, after 1983, I have not listed anything because development is everywhere. If you just Google it, you can find it everywhere. The development of plant tissue culture is there. So in case of carrot, in case of production of medicine, in case of crop improvement, you find plant tissue culture everywhere. But I have not listed, as I told you, first is always listed out. Anyways, this is all because of the characteristic feature of the plant cell, that is totipotency. This is the ability of the plant cell to produce the whole plant from a single cell. That is known as totipotency. That means if you just keep a cell in the culture tube, then cell has the ability to produce a whole plant with the help of a sexual reproduction named as mitosis. We all know that. So when they grow it, how does it look like? If we give the tissues in vitro, can you see that PLB? They form round structures. And we don't call them callus. We call it protochrome-like bodies. PLBs we call it. And they developed into a full-grown plantlets. We call it, the smaller plant body, we call it plantlets. So that's how it gets developed inside the culture tubes. This is very much with the aseptic condition. Now for all this, what exactly do we need? We need the explants to use. It could be the young leaves, it could be the roots, stems, seeds, or after the formation of PLBs, the callus itself. Once they grow inside the tubes, then we can transfer them also, which we also call as subculturing, so that we provide more nutrition to it. And then they simply grow it into a full matured plant body. That's very easy. So what are the features? What can we get out of this plant tissue culture? Soma-clonal variation. Then we can conserve the germplasm. Then we can go for breeding with the mutation. Then we can definitely try fusion of the protoplast. Protoplast from the different cells. It could be interspecies, it could be intergeneric, we can try. Then we can start with embryo or the meristem culture. Then we can produce disease-free plants. Then we can go for farming, molecular farming. Then we can be the engineer of genetic genes. Which particular gene can be inserted. And the production of secondary metabolites. That means after studying what secondary metabolites are present, what alkaloids, tannins, etc., what are the components of the plants. After studying that, if we require the particular secondary metabolite, we can produce that. So these all are with the help of O. That's the features of plant tissue culture. So for that, we exactly need media components. These are nothing but the inorganic salts, which could be major or minor elements. Depends how much we require. Major elements, as we all know, these are the major elements, as you all can see in the slide: calcium, magnesium, phosphorus. These are the, and minor, molybdenum, zinc, cobalt. These are the minor elements. Apart from that, we need sugars, starch, and cellulose. Moreover, we need vitamin B complex and the growth regulators, the auxin, cytokinin, gibberellin, ascorbic acid, yeast extract, etc. And this media, it could be a suspension, liquid also. But if we keep it in the suspension culture, what happens right at the beginning is explant needs a rest to absorb the nutrient. So in that case, we supply it, or we solidify the media with agar. And we all know the source of agar, at least students, you all have studied, so I'm not going to tell you. You have to Google it if you don't know what has come. Agar has come from what, okay? So that is about the media components. I'm talking about the particular media here, right? On top, you can see Vacinanvent, Nutsensi, MS, Orchumex, OMM. These are the name of media. In earlier life, or the beginning of the plant tissue culture, they used to combine all these salts. Components, would you see in the left corner, there's salts they used to combine and prepare the media. But right now, again, the biotechnology, the improvement, we get ready-made media that's available in the market. So they come with a name that is 1, 2, 3, 4, 5 names I have given here: Vacinanvent, Nutsensi, MS, Orchumex, and OMM, that is Orchid Maintenance Media. So these are the different components that forms the particular media. And that is also easily available in the market. So you can see that activated charcoal is present in the Orchomex media and OMM. Because I'm saying being particular with activated charcoal, because I'll explain it when I'll take up with the case of orchids. Because activated charcoal is very much important for the orchid nutrient media or the tissue culture of the orchid. So this is the laminar air chamber, what we call it, which has the presence of different components like filter pad, HEPA, high efficiency particulates air filter, that filters the air, and the fan sucks the air through the filter pad where the dust is trapped, and it helps to overcome the contamination. So if you see the earlier slide, do you see the instruments? This all needs to be sterilized. Okay. So this sterilization is done with the help of autoclave. That's the top view. And this is the outer view of the autoclave. Autoclave helps in sterilization of the nutrient media as well as all the glass goods, all the goods that we're going to utilize during the process of plant tissue culture. As because this is aseptic condition, we have to sterilize everything. So how it works? It gives extra temperature. What happened to it? Okay, so it gives extra temperature so that all the germs are killed and maintains a pressure of 250 degree Fahrenheit, that is 121 degrees centigrade, with 15 pound of pressure per surface inch. Yes. So after that, after keeping it there for 15 to 20 minutes, everything is sterilized and we directly bring them into the laminar airflow. And after bringing it to the laminar airflow, we do the inoculation of the tissue culture, and we have to keep it in the rack. So these racks are fitted with the tube lights because we have to maintain a certain heat in the room temperature, in the room. So for that, we maintain the temperature with the help of the tube light, and we provide the light with alternately with 8 hours of photoperiod. We call it the giving photoperiod of 8 hours. We provide the light for 8 hours. At present, again, the technology or improvement in biotechnology, we get this rack also automatic. Like for this many hours, the lights are on, and the rest of the hours, the lights are off. We get that also. But in Darjeeling Government College, we had this kind of tube light fitted racks. Now, this is something designed. Well, this is somewhat like laminar airflow, but we call it, I gave it a name, inoculation chamber, because we can work with this also. And this has been manually designed with the help of one teacher's idea. I have designed this table, covered it from 3 sides, and you can see the holes there. These are holes. Now what happens? From below, we keep a heater. Above heater, we keep the boiling water, and the steam passes from these holes, and here we work the tissue culture. And why have I designed this? Was when I was working with village people during women scientists. As a women scientist, when I was working there with the village people, we could not afford to buy laminar airflow. So in that case, we designed this and we learned plant tissue culture with the help of this inoculation chamber. So how about the HEPA filter? So what we did was we fitted with exhaust fan from the back, and it used to pull all the airs from there, and we used to keep every other things in front, and with this steam and with the help of spirit lamp we used to do the inoculation. Yes, of course, there is the success story of that time also. And a normal place for keeping the glass goods and the nutrient media that is required. We can be the entrepreneur if we start this. Then the normal man-made, homemade, rather I should say, homemade, the rack and tube lights were fitted with that. So after that, whatever it is, however it is possible, methodology, what methods should we follow? Collection of the explant. That was very much easier if we go to the villages, because orchid seeds are, so I'm giving you the case of orchids now. I'll start with the giving an example of the orchid, tissue culture of the orchid. Now I'll start with that. To bring the explant, because once they are in the forest, they find this green seed pods, they're about to rupture, which gets destroyed in the environment. So they can collect it, bring it home, and they can do the tissue culture of it. So after that idea, we've developed this. So they can collect the explant, then they can inoculate in the nutrient medium. As we all know how to prepare the medium, then they have to maintain it in the culture room. And after they are well developed, they can take the process of hardening. Hardening should be done at the beginning in the greenhouse, then only in the open field. So that's the basic methodology in tissue culture. So in tissue culture, we find 4 different stages, from explant to a mature plant. So explant is a very minute object, very, very small. So what we can see just now in the slide, it's of the orchid seeds. Now I'll come to that. Why have we taken orchid seed also? So that is why I said it has to be agar solidified, because the seed itself, it blows away in the air. It needs to be settled down in the nutrient media. And the initiation, swelling, etc., that takes place within 7 to 10 days. Then comes the multiplication steps, the most fascinating phase in the tissue culture. When you see a small micro part or the tissue from the plant body, and you keep it in the nutrient media, and after within 4 or 5 weeks, you find multiplication of the same with the help of asexual cell division. Then comes the next stage, 8 to 10 weeks, rooting of the same plant. Then we can get it hardening of, it's normally after, it depends, because I haven't given the particular time factor there, because it depends upon the media. Now you need to subculture it, you need to change the media, you have to keep on watching them. That is why after how long, I'm not particular of it. Okay, but in case of orchid, it has taken a time period of 3 to 4 months. So after that, it's ready for acclimatization or hardening off. That means going to the greenhouse. So what technique we should definitely know is handling the tissue and the culture in total sterile condition, and knowing the medium. Seeing the medium, we'll have to know, do we need to change the medium or not? So the practice makes man perfect. We can expertise accordingly. So what are the advantages? So when we propagate them, we call it propagation. And this tissue culture is the propagation from a micro part of the plant body. We call it micropropagation. So this enhances the rate of multiplication. And why do we need to enhance the multiplication? Because there's a constant demand of the new varieties or the improvement of the crop or in the ornamental industry. So there's always demand of micropropagation. And in case of orchids, the seed of the orchids in natural, they germinate by accident. That is, if in case they come across with the suitable partner, mycorrhizal association, then only they germinate. Otherwise, there is no life for the seed of the orchids. That is why it was more important to save the orchids. And if we see it in the Eastern Himalayas, orchids are being very, very rare and endangered, that they're being extinct now. So total number of orchid species within India is that much number of species. And Eastern Himalayas, it has 72%. And rare and endangered species here is 23%. So out of this, if we can save some of them with the help of tissue culture, that'll be a great effort. So if we see the percentage depending upon the status, that is rare, ornamental, endangered, and others. In the previous slide, you must have noticed rare was 23%, that is all together in the Eastern Himalayas. But this graph, what we are seeing just now, is only in Darjeeling Himalayas. So we have more of the scarce and rare orchids. So that is why it's very important for us if we do tissue culture of orchids here at least. That is because in Darjeeling area, there are the use of orchids in ethnobotany. There is huge use of ethnobotany. In case of ethnobotany, they use all the orchids without knowing that it may be produced in natural habitat, it may not be produced in natural habitat. They're just taking it and using it. And it has the different uses. As you all can see, this side, the genera of orchids, and these side are the medicinal uses. We have different. So the common diseases like headache, see, Silogeny corymbosa. That Silogeny corymbosa you find everywhere in Darjeeling, but that is also being like, it's, you don't see it like earlier it used to be everywhere. Now you hardly find corymbosa everywhere. You have to go and look for it. Those white orchids. Silogeny. Then for cough, urinary infection is Silogeny ovalis. It has already been limited to the grows. Silogeny ovalis, you don't find it anywhere now in the natural habitat. So similarly, other orchids also, they have medicinal uses and which are being used very much by the practitioner. So it's our duty how we can save it with the help of plant tissue culture. That is why I've given you the example of orchid today. So Rhynchocelis retusa, they are already extinct, or they're already rare cases. They cure the diseases like rheumatism, asthma, nervous twitching, cramps, vertigo, and even in the kidneys. So once we come to know all these utilities, we definitely use it. So when we use it, why not replace it? Sustainable utilization. That is what we're doing with the help of tissue culture. We must have, at least after being the educated people, we must realize how to utilize, how to have the sustainable utilization of whatever we use. We must learn to give back to the nature earth. Otherwise we suffer like Mother Nature has the ability to stop us as we've been stopped right now. Okay, get back to the orchids. The particular is Dendrobium. I went with the particular one, Dendrobium. And there are 1, 2, 3, 4, 5 Dendrobiums which are used here practically: Chrysanthemum, Densiflorum, Jenkinsea, Longicornu, and Dendrobium nobile. Dendrobium nobile is even called as king of the orchids in Himalayas. Yes. So we can see the different uses or the medicinal uses of Dendrobium. Other than that, this primulum, this is ornamental, and these are being very, very much rare. This is rare and endangered species: Fimbriatum, Crepidatum, and Chrysanthemum. So we can do the tissue culture for all these Dendrobium species, and we can say that this is the intervention in biotechnology. So this Dendrobium nobile in particular, I give you the example because this plant, this is the description of the plant, which you can find anywhere, mainly used for night sweats, fever, anorexia. It is used as analgesic, antipyretic, and a tonic to nourish stomach. And the recent pharmacological studies have demonstrated it as antitumor, antigenic, antiplatelet, anti-inflammatory, aggregation, antimutagenic, and the most important, why am I giving you the example of Dendrobium nobile, is immunoregulatory activities. It's because of this particular factor. Why not? Plant tissue culture of Dendrobium nobile should be at first immunoregulatory activities. It has the properties. So now we can imagine how much plants will be uprooted from the natural habitat. So apart from that, the seed does not germinate. If it has to germinate, that is by accident, because it needs an association. We all know that. So need of orchid micropropagation is mainly because of these 4 points. As you can see, this demand, overexploitation, production of new varieties, and they do not germinate in natural habitat. So when we give it in the nutrient media, the seed, you can see in figure 1, the seed, they are without endosperm. It's a naked, figure 2. Can you see that? The seed does not have the endosperm. That is why they are unable to germinate in the natural habitat. That is why they require an association. So that is why that's the fate. It's, if they have to germinate, it's the accidental case. So what we can do is we can preserve them, we can conserve them. We get numerous seeds in a seed pod. We can bring it to the lab. We can do the aseptic culture, and that's how they grow. Developmental stages of the orchid seed: swollen embryo, they swell up once they get the nutrient medium. Then I said protochrome like bodies, protochrome stage. Then they start giving the shoot and the primitive root and forms the plantlet. See, from the protochrome you get a root, with the root a small plantlet, and they're ready for the acclimatization. Now the percent of germination in case of which particular media? Particular media or OMM, Murasican scoop, and Vaccinan went. OMM, Orchomex, was not available. So if you all remember the media composition, OMM had the presence of activated charcoal. That is what required in the germination of seeds of orchids. So OMM has the maximum percentage of seed germination in comparison to Murasic and Skook, and Vessin and Vent was the least. So if you see the in a graphical manner, the highest was of course, OMM, that is orchid maintenance media, and the lowest was fencing and wind, and the middle one, average, was Murasig and Skug. Okay, and after that they have germinated half the strength. That means now give them a lesser amount of the nutrient. With the lesser amount of the nutrient and different combination of the hormones, with 5 mg per liter of BAP, we've got 99% germination. That means they are full grown plantlets which are ready for acclimatization. So that's how now we can see it with the graphical representation also. With higher the dose of BAP, more is the growth of the plant. That means not only the growth, it's the plantlets itself are very healthy growth. So need of tissue culture in orchid seed is importance of orchid diversity. As we have come to know, the diversity of orchid in our area then protect depletion of the orchid wealth. And this technique helps to increase the seed germination. Otherwise, seeds are destroyed. So this is fascinating. This is easy as long as we work, but it gives some problems everywhere. In every field we find some problems. So what happens? Culture gets contaminated. What could be the possible causes? Infected, improve sterilization method. It may die off, change the sterilizing agent. When you are sterilizing, maybe they are dying off there only. They are died there only. Review the sterile technique, change the media. So that's how we can work with that. So how it is relevant to this? That means what are the things that we can do it? Media can be optimized. Okay. Then medicinal important varieties, horticultural varieties, we can improve it. Disease-free plant, reintroduction of rare orchid varieties into the natural habitat, we can give it, and we can get the genetic variation in the plant itself. This, as because I have scanned from the book, it's just a flow chart. That means orchid habitat from there, how we can get it, how we can evaluate the orchids, then how can we give it, which particular orchid to give it for the tissue culture, and which one you may choose it there, which stage, and you can give it to the growers, and we can give it to the gene bank also, and that's how we can keep it with the help of conservation technology and all. So successful cultures within India was seed and seedling explant, embryo culture, regeneration in the root culture and the shoot culture with the help of axillary and lateral bud, leaf explant, anther and pollen culture, and endosperm, even the endosperm culture. That's how these are the success stories in India. So the particular application in the genetic engineering is not possible without tissue culture. I mean, not possible in the sense, in the development of the plant tissue culture. Okay, genetic engineering is important, but how do we do it? After the sequencing of the gene, once we find that this particular gene is required, then we cannot do it directly. It has to be inserted into a particular cell, in a particular cell of or then it has to be grown with the tissue culture only. It needs nutrient. So regeneration of the whole plant from a single cell, genetic engineering requires plant tissue culture. And efficient regeneration system are required for commercial success of the genetically engineered products. Moreover, plant tissue culture, after that, we need more efficient regeneration system. That is what the development of biotechnology. And the germplasm conservation. Now, once we get it, we have to conserve it. How do we do it? So loss of this, after getting the plant tissue culture plants, we have to conserve it. We must have a gene bank. So that is definitely required there. So that does not get stressed with the other natural factors. So it has to be conserved with within the germplasm conservation. And We can utilize tissue culture in embryo culture. That means to break the seed dormancy and test the vitality of the seed. So how long will you keep the seed? You test it in the plant tissue culture, and that's the molecular farming you can start with. So these are the things that we come across when we handle with tissue culture. Genetic transformation, the most recent feature of plant tissue culture. Transfer of the gene with desirable trait, that is genetic transformation. You can become a genetic engineer. So successful introduction of this agronomic trait or the desired trait in the plant, that can be done through genetic transformation. So these all are the biotechnological intervention. And the protoplast fusion. Fusion of the protoplast, that means these are molecular level techniques inside the tissue culture. So develop successful development of the hybrid plants. They are the stories of citrus fruits, which we can study further, we can learn further. These are really interesting things once we start with tissue culture. So moreover, citrus fruits are very much in demand. It's going to be because it provides vitamin C, what we required just now. So protoplast fusion has developed that also. And the haploid production to produce the homozygous plant in a very short period of time. And it helps out in conventional breeding. So that's how we get the desired trait of the plants, that is drought tolerant, disease free plant, and the long leaves and short plants with the flowering, with the flowers more matured earlier, and it lasts for 3 to 4 months. So all those development through tissue culture. Okay, somatic embryogenesis and organogenesis, that means tissue develop into differentiated embryos. So you don't have to wait for sexual fertilization. You're just doing it in your hand with the help of tissue culture. Establishment of the unorganized cell or the callus, and from there you're developing a new plant. And great promise of tissue culture is in pharmaceuticals. Right now we need it very much, because the secondary metabolites which are present in the plants, if we can extract from each and every plant, because every plant has the medicinal properties. So if not each one of, if 10 out of 100 learns this tissue culture technique, then there will be success in biotechnology and tissue culture. So pharmaceuticals means preparation of the medicines. So instead of using a chemical components, so develop more, because we need more. So that's how these are the applications of tissue cultures. So the current and the future status, it says that it has evolved very much, but still it's a growing process. It's an increasing demand in a productive system in several countries. So taking care of that several countries, charity begins at home. So within India, within in our own state, we can start ourselves also. Thank you all for watching this, and I hope everyone has enjoyed about it. I hope this was you've got basic knowledge, and we can start with this. Thank you very much. Thank you, organizing team. Thank you, Didi sir.